Today is going to be a short little video on what we do if, when we're running an experiment, we have a confounding variable that we know about and that we want to compensate for. So completely randomized design is the simplest, but sometimes that's just not going to work. So what we would do is we're going to divide our, just like when we sampled, we created strata, which were groups that we knew were all the same, and then sampled a little bit out of each group, so we made sure that we got the entire range of the population. A block is a group of experimental units that are known to be the same, and so we're grouping them together to get better results. So in a randomized block design, instead of just taking the population and putting half in one group and half in another group, what we will do is we'll separate people into blocks. And then we'll compare, we'll do treatment to the two different blocks and then compare. So, and I know this drawing isn't super clear, so let's look at a better one. We'll look at a better one in one slide. We basically form blocks based on the most important unavoidable source of variability, a potentially confounding variable among the experimental units. Basically what we want to do is we want to control what we can, block what we can't control, and hopefully randomization will take care of the rest. So read the example on page 246 of your textbook. It's highlighted in blue. You're doing the laundry. You can pause and get your textbook and come back. You're going to do the laundry, and you're going to test a new detergent on clothes that require hand washing. The problem is that light-colored clothes do much better in warm water, where dark clothes it doesn't matter. And so how are we going to handle that? Handle that with the hand washing. How are we going to create an experiment to see if in fact this works better? So what we do is beforehand we'll take our laundry here and we'll divide it into light colored and dark colored clothing. And then we are going to randomly assign within the blocks. The key is that assignment to blocks is not random. Assignment of treatments within the blocks is. And so then we would do the light colored clothing in cold water and hot water and compare the cleanliness, and we would do cold water and hot water and compare the cleanliness. And this would tell us whether the detergent worked better in warm or cold water for both light colored clothing and dark colored clothing. So a block is very similar to a strata and sample, but we're dividing our experiment into things that we know are going to be confounding. And we're going to do more of this in class. So we block to make the experiment more accurate. Or we block to reduce the variability of the results. The other thing we can do, whoops, that went the wrong way, is we can do what's known as a match pairs. And a match pairs is different than a block. Block is where we separate the population into two groups or more, two or more groups, and then randomly assign treatments out of those blocks. A match pair is where we give the exact same treatment, or we give two different treatments to the same experimental unit. And then all we're doing, then we're getting even better results because any variability is negated by that individual unit. And sometimes a pair is a single unit that receives both treatments, but not necessarily. Pair can be another way, can be done another way. An example of this would be if you wanted to see if tread lasted longer on a kid's shoe. Well, you can't give two different kids two different shoes because the kids may behave completely differently. And so what you could do is you could put the new tread on the left shoe and the old tread on the right shoe, and do that for a whole bunch of kids, and then at the end compare the shoes to each other, because you know they got the same amount of wear and tear because they're being worn by the same kid, and so you knew they were all treated the same, and any difference between them must be in the treads. This is what's known as a matched pairs design. There still has to be randomness in the assignment of treatments. Here it would be whether the right foot or the left foot got the new or the old treatment, or in this case, sole of the shoe. And we'll look at more examples of this in class. We'll actually do one of these real briefly in class, and then we'll also do more work on this. So going back, we have two different, more precise types of experiments. We have blocks, where we split the population into groups that are the same and then assign treatments out of those groups, or match pairs, where we match things up that are the same but give them different treatments. And I know they sound similar. It's going to be a lot clearer in class when we do a few examples of these. Also, you may want to read through this section of your textbook just to kind of look at more examples of the difference. So in this section, we learned that we can produce data intended to answer specific questions by experiments. We talked about identifying the experimental units and how that is the smallest unit to receive a treatment. We talked about how we designed experiments. We talked about how important random assignment was, how important control and replication were. We talked about what a double blind is. Made sure we knew what a treatment was. We talked about a placebo and the placebo effect. And then we talked about blocking experiments and match pairs. One of the biggest ways to tell the difference between a block and a match pairs is the answer to match pairs will always be the difference between the pairs, where the blocks are comparing two different populations. So, and this will make sense later, but if we had blocks, we'll end up with two different averages or two different proportions. But in match pairs, we would only end up with one, which was one average, which is the difference. And so a lot of times, especially on an AP exam question, if you're confused about whether it's a block or match pairs, look at the results. The results will often tell you which one of those two experiments it was. And we'll do more of these in class tomorrow. I know this is a relatively short video. That's fine. If you want to, read over the section of the book. And I will see you guys next class.